Hello, students. Welcome to our Hardy-Weinberg online lab. I hope that your first couple days of virtual school have gone well. I miss you very much. So does Rhino Man. And so let's figure out if the rhino population is evolving, and let's do that with Hardy-Weinberg. So this is from the AP College Board. We're going to go down here, and you'll see that it's investigation 2. So I'm going to click on the student version, and you could read this. I made a handout for you that kind of steps you through what was in this lab manual. And I made that handout just by following the directions in here. I had to look up a few things for Google Sheets that were different commands than Microsoft Excel, and everything seems to work. So I'd like to thank NJ. She gave me the idea that to let you earn some points, you could write up this lab, or you could write up the BLAST lab that we'll do later on, and you can choose to do like a CER, a claim evidence reasoning, on either of those. You do not have to, though. I'm not going to make you write up these virtual labs. So if you are happy with your grade in the class, or if you are working on the AP classroom questions or something else and don't think that's the best use of your time, you're not going to have to do it. But NJ, you can do it. All right, so what does the Hardy-Weinberg lab look like? Here is the end goal. This was my sheet. Watch this. So it's going to have the frequency of the dominant allele, frequency of the recessive allele. Here are the alleles, right? And so this would be haploid, and then this would be the diploid individual. So there's 16 individuals here in this population. Let's change this to 0.6 frequency for P. That would be 0.4 for Q. Looks like my formula got jacked up there. And you saw how it changed the sheet, right? And so, well, here, look, this is just randomness, right? But A and B, so there's 10 alleles for A and 22 for B. I think I pressed the function that messed up the sheet. So we will make a fresh one together. But you can press Control R or Command R, and it should constantly refresh the sheet and make it recalculate random values. We'll go through all that. What is the point of this? What is the Hardy-Weinberg equilibrium talking about? And hopefully you've got an understanding of it by now, that is showing that the alleles in a population, the frequency of them are going to remain constant if there's no evolution occurring. So essentially, it is the null hypothesis that Shreya brought up in class, that nothing's changing. And we know that this is going to be broken all the time. There's always some types of evolutionary forces at play, whether it's sexual selection, natural selection, some type of fitness level. But what's cool about this is we can use it and we can see the deviations in the change, to see the rate of evolution or how quickly it's happening. We can model something like COVID-19 and see if we change the selective pressure, what might happen to it. So it's a very useful null hypothesis for us to do experiments on. And I'm working on finding some chi-square with Hardy-Weinberg for your Thursday lesson. There are online simulators. This is one I've used in the past, and that you can model different things and see how changing the sample size changes the frequency of the alleles. You can see that some alleles might get lost over time. So let's change the population size here from 50 to 500, right? You said go. And look at that. It's going to stay a lot closer to equilibrium, right? Which makes sense. The larger the population size, the less chance that an allele will get lost. Here, it still happened in population of 3. You can see it takes longer. So these types of models are always fun to use online, but it's not as good as making our own, because then we feel that sense of accomplishment, right? I always like when Lisa's like, I accomplished this in lab. So let's do it. Bear with me. I'm going to run through it. I've already restarted this video a couple times, and so we're going to just rock it out here. If I make any mistakes, we'll go back. You could turn the video off and just try it from the sheet if you like, but if you want to go with me, you can. All right, so I'm going to call this Hardy. Oh boy, give me Hardy-Weinberg lab in class example. I guess it's virtual class 2020. I'm going to make this, well, let's make it 125% so you can see. In A1, I'm going to put John Gibney Hardy-Weinberg modeling. In B2, I'm going to put P is equal to the frequency of A. In D3, you're going to say Q is equal to frequency of B. All right, this is right from my sheet right here, and it's also right from the lab manual. Okay, so let's see. We are now on to D2. And in D2, I'm going to put in a random number between 0 and 1. I'm going to try 0.1. In D3, I'm going to say equals 1 minus D2. First formula. So what is this saying? It's saying, okay, if the frequency of A is 0.1, then the frequency of B is going to be 1 minus that. So if I want to change the frequency of the alleles, I just need to change whatever P is. And look, it should add up to 1. P plus Q equals 1. Okay, we are on to E4, and I want to put in the word gametes. G4, I want to put in the word zygote. G3, I want to put in the word number of each genotype. Okay, H4, I want to put AA. I4, I want to put AB. J4, I want to put BB. So we oftentimes represent dominant and recessive alleles by capital and lowercase letters. It doesn't really work for Excel, and it's not really how we label things with fruit fly genetics. You saw that with VG plus being the wild type and VG being vestigial. That's how we really do it. When we talk about sickle cell, we talk about AS, and then SS. That way you don't confuse. Like if you're drawing the S's, it gets confusing. So here, A represents the dominant allele, B represents the recessive allele, and AB would be the heterozygote form. All right. So let's model this sexually reproducing population. In E5, here comes our first formula. Okay, equals if a random number between 0 and 1. That's a pretty cool function, right? Look at this. Do I feel like Mr. Glenn's? Or equals D $2 A B close parentheses. So you can check that out. You could copy and paste it from my sheet right there. So what is that saying? If the random number is less than or equal to the value of P, then it's going to put an A in the cell. If it's greater than the value of P, it's going to put a B in the cell. So it's taking a random number between 0 and 1, and it's saying, okay, 0.15, right? If you're 15 years old, 0.15. If it comes out as a 0.15, it would give an A. Here it came out, and it doesn't tell you the number. We'll say it's 0.6. It came out as B. Press Control R. It's going to make it constantly, it's refreshing that cell. I guess Command R, I believe, on a Mac. And you see finally an A popped up, right? And A should pop up less. It's only going to be around 20% of the time. That's the frequency of that allele. Okay. So it's saying, class, any questions? All right. In F5, we're going to put the same formula, right? Because here's one allele from mom, and then we also need one from dad. So, oh boy, why did it do that? All right, I'm just going to retype it over. Equals if rand, oh boy, equals D $2. I'm sorry, I don't remember what the dollar sign means right now, but it's important. Okay, that is the two haploid sex cells. Now they're going to fuse together to make the baby. So, oh, hold on. All right. Drag the formula down from E5 to E20. So here, go lower right-hand corner, drag that down to E20. Bam. So that is modeling 16 different individuals, I believe. Drag it down to E20. Okay. Here we go. For G5, now I don't know how to pronounce this. Concatenate, we'll say, equals concatenate. What that means is, look, it tells you, append strings to one another. That's pretty cool. So it's going to add the two cells together. It's going to add E and F to make the baby. So we're going to say E5 and F5, and it gives you little boxes, which is kind of cool. Make that baby. Okay, get it, Hisham. There we go. Let's drag that formula down from G5 to G20. Here we go. All right, there are some wonderful babies right there. Okay, now we're going to go over to H5. H5 equals, this is kind of the number of homozygous dominant, heterozygous, and homozygous recessive so that we can do some Hardy-Weinberg, all right? So we need to count all the homozygous dominant, so 2 A's. So we're going to say equals if G5 equals AA, give a 1 or a 0 if it doesn't. All right. We're going to drag that down to H20 now. So we had 1 homozygous dominant out of the 16-person population. I'm going to shrink these cells. Oh, that's a little too much. I'm going to shrink the cells a little bit because my head is going to get in the way over there. Thank you. All right. Imagine the fun if we're doing this in class. Okay, equals. It is fun. All right, now this one's a complicated formula, right? Because you need to count if it's AB or BA, right? When a cell is expressing a gene, doesn't care if the dominant allele is first or recessive allele is first. It's just expressing the gene, transcribing it, and making the protein. So we're going to say equals if parentheses G5 equals AB, give us a 1. Then we're going to say if G5 equals BA, give us a 1. If not, give us a 0. All right. So let's see how many heterozygotes we have. Okay. All right. So we have more heterozygotes right there. Okay. So we have, let's see if it worked. Oh, yeah. That one, that's a heterozygote. That's a heterozygote. Look at this. Go, Nico. We are getting the right stuff. Okay. Let's do the homozygous recessive, which is right what we would look at with the Hardy-Weinberg equilibrium. I sent Mr. Kelly my Hardy-Weinberg video the other day, and I don't know if I explained it well enough, but right, we're always going for Q squared because we can't look out and tell the difference in a population between homozygous dominant and heterozygous. So we're starting with Q squared, and then we solve for Q, go to P, solve everything else. All right, I lost my mind a little bit. J4, we're going to put equals if parentheses G5 equals BB. Homozygous recessive gives us a 1. If not, give us a 0. There we go. Okay. Now, because I put the frequency idea, I should get a lot more of these, and it looks like it. All right, let's change this to 0.3. Oh, it changed all my values. Pretty cool. Okay. Now, it's hard to look at this data and get anything from it, so we're going to do some summation things. We're going to go to C21, and we're going to write sums for each genotype. Then we're going to go to H21. We're going to do a formula. Equals summation of H5 through H20. You could go up and just drag the bar. Let's make sure that covers all of it. It covers the 16 people. Yes, it does. So we have 4 homozygous dominant individuals. Now we're going to do the same over here. I think we can just drag the formula across. Let's make sure sum I5 through I20. That works. And this should be sum J5 through J20. So this should add up to 16 individuals. So what is this saying to us? We have 2 homozygous dominants, 7 heterozygous, and 7 homozygous recessive individuals in this population. Okay, let's now go to H23 and let's type the letter A. Whoa, look at that. When we typed in that A, it changed everything, right? It had nothing to do with the formula, but basically when you type in a cell, it's going to recalculate all those random values, which is pretty cool. All right, and if you want to force it to recalculate random values, it's Control R or Command R on a Mac. All right, that was H21. We're going to go to J23 and we're typing B. In C24, we're going to type in the words number of each allele. In H24, we're going to do a summation of all the A's. All right. We're going to count the summation of all the A's in H21. And here's the trick, right? We need to add that twice because they are homozygous dominant. And this goes back to the question from that exam that I showed you where you had to calculate the allele frequency and you had to look at the dominant B or the recessive. So think about it. All these individuals that are homozygous dominant, that are AA, are going to have two copies of it. So we need to add it twice, right? So we're going to H21, H21, I21 one time, right? And I21 one time works because we know that they have a copy of an A or a B. So we summation that. I should practice this much for every lesson. This is fun. Equals sum I21, J21, J21. So let's think about this one. I put that in the wrong box. Come on, Gibney. We're going to see if we can copy and paste it over here. Let's see if it worked. Yes. All right, so let's see if this works for the B's, right? So we're going to get one of the I21 here. So one of these 8 heterozygotes. So we know that they have 8 B alleles. And then we know over here that there are 8 homozygous recessive individuals. So we know that they're going to have 16 B alleles. 16 plus 8 is equal to 24. So everything is working out so far. All right, let's do another control R, make it recalculate. It is doing great. Okay, and now it says go to H25 and type in P. Go to J25 and type in Q. Done. 2 sheets, done. Okay, we're now going to go to C26. We're going to write in allele frequency for next generation. We're going to go to H26. We're going to type equals H24 divided by H24 plus J24. So what does this mean? This means that the allelic frequency of the dominant allele of P, which in this case is A. Woo! Say that twice, 5 times. Right? The frequency of the dominant allele is going to be 9 divided by the total number of alleles, just like we did in that problem. And so P for the subsequent generation will be 0.4. And then the Q for the subsequent generation is going to be equal to, you could just say 1 minus that, I believe. Let's make sure it works. J24 divided by H24 plus J24. Oh, and then recalculate it. Yep, it adds up, right? So look, P and Q for the next generation are 0.3125 and 0.685. Here is 0.3 and 0.7. So is the evolution happening? Yes, there's some changes, but it's pretty close to the first ones here. So we could do some type of chi-square or null hypotheses. Okay, you can make a graph for that. So let's see what we can do here. All right, this part I did not practice as much. Highlight H4 through J4 and press Control. Highlight H21 through J21 and then press graph. H4 through J4. Now I'm going to press the Control button. Then I'm going to press H21 through J21. I'm going to insert a graph. I'm going to insert a column graph, non-stacked. Move my head over here. I got it, Google. Column, non-stacked. I'm going to go to Customize. I'm going to edit the title. So chart title, genotype, frequency, and next generation. I'm going to go down to x-axis. chart and axis titles. Okay, chart title, I'm sorry, horizontal axis title is going to be genotypes. Vertical axis title is going to be number of offspring. Go back to the data part of the sheet and click on series. And click on the appropriate cell for each series. So for H21, the label, add labels should be H4. For I21, the label should be I4. And for J21, the label should be J4. Okay. All right, cool. So, pretty cool. You've made the model. All right. I'll let you do the part at the bottom where you add multiple generations. All right, because this is already getting a little long. But let's see if it keeps changing. All right, so here is my nice graph. And I'm going to go over here and I'm going to click Control R, recalculate. Oh, my goodness, look at it go. It's showing the change in the allele frequency. So we could always look and see, right, if the changes from the numbers up here where my mouse is, 0.3 and 0.7, then evolution is happening. So you can model a lot of different things. You can model bottleneck effect. You could, you could model any of the forces that go on here. Let's change this to 0.5. Let's see if it comes out pretty even. Right, and so it does, right? Heterozygotes. Let's do it again. Cool. It shows a little bit of randomness, etc. So you can stop it there. Let's see. I don't think it's not going to let me add a sheet while I'm doing this screencastify. So you can follow the rest of my directions there, and I'll post these with the video. Okay, keep rocking and rolling on your virtual schooling. Take care.